everyone, my name is Clara. I am a grad student from the Editor Lab at UCSD. Today is the CM4AI internship day 2, and I will give you an introduction on how to evaluate LLMs in gene set analysis. You've already heard from yesterday on how we use cell map for interpret systems that are important for drug response. Basically, a cell map is a hierarchical network containing assemblies of proteins that interact with each other across different biological scales. Here is showing an example of cell map. At the bottom is the whole cell, and as you move up, you are seeing more detailed organization of proteins. For example, you are moving up one layer to nucleus, and there's systems involved in cell cycle, and within cell cycle system, there's DNA damage repair systems. And you can see there are highlighted protein systems here showing in the slide, and those are the systems that are identified as significant for drug response that was published earlier this year in Nature Cancer. And here I'm pointing out one of the systems here with 15 proteins. So now we're raising a question is how should we interpret the gene set function of this group? So what do I mean by interpreting the gene set? I specifically mean these bullet points listed on the slide. What are the functions of each protein? And is there any evidence that they work coherently as a group? And do I believe that they work together in a group? If yes, then I would like to annotate this system. And it would be better if I can have a succinct report on how I reasoning through the functions of genes to give this name. So to check all those boxes, there's a lot of time and labor needed, especially if we need to scale up on the number of proteins or the number of systems that we want to interpret. So given this need, there's another question is raised up, is that can we use large language model to help us with this task? If you don't know what exactly is large language model, or LLM in short, I believe you all heard of or used ChatGPT at some point. So the model behind ChatGPT is an LLM. So it has seen massive amount of public data and literature, and it is able to generate human-like text based on the embedded knowledge it has seen. So knowing that they have the ability to synthesize information, we propose that they can be used for this task to help us interpret the gene set inside of the cell map. So with that idea, we developed an automated pipeline to query LLMs for gene set name, the analysis that support its name, and a confidence score that evaluate the name, which I will go into details on individual sections. So the key step in our pipeline is the prompt. Prompt is the text input to guide the LLMs in generating a response. I will go through the structure of our current prompt, and you will have an idea on how to structure your own prompt during your hands-on experimentation in today's challenge. To break it down, first is to specify the role for the LLM in system content. Why this is important? So telling the role helps the LLM to be more focused on the certain context in its response. And we saw that the result actually aligns better with our expectation. So here, the context is molecular biology. So we are telling it to be an efficient and insightful assistant to molecular biologists. So next, we follow a technique called chain of thought prompting. This strategy involves in guiding the LLM step by step through reasoning process, helping the LLM to understand the goal better, and it can be better at solving the problem. For example, we here ask it to write a critical analysis for the biological processes, and we specifically telling it that after you complete your analysis, propose a brief and detailed names for the most prominent biological process. So we implemented chain of thought prompting again in the instruction on the confidence score. So here we're introducing a new concept that's a confidence score, basically allowing the LLM to self-reflect its answer on how accurately the chosen name represents the functions and activities of the system. And we define the range of confidence between 0 and 1. So this is additional layer for LLM to self-assess, and it can indicate the model's certainty about its analysis here, especially in how it names the gene set, and give us a hint on how much we can trust this model. More importantly, I would like to emphasize here that is, we ask it to do not name a gene set if it figures that these genes do not work coherently. In other words, there's no prominent biological process. Why this is important? Why we don't want it to name a gene set? So in real life scenario, there might be artifacts, contaminations, false discoveries, and during the experimentation or downstream data analysis. So it is more important for us to have a hint on potential noise and data. We don't want the LLM to hallucinate a name for any given gene set. So here we specifically specified in the prompt that we wanted to name a process system of unrelated proteins and give a 0 score if it finds that those genes do not work coherently, or if the name cannot describe the functions of the gene set. And keep following the chain of thought, we give it more guide on how to refine the output, including being concise, avoid unnecessary words, be factual, not editorialize. So beside the chain of thought, we use another strategy. Here it's called few shot prompting. What does that mean is it gives the model a few examples. Here we give one example of this task we want the model to solve. So it can help the LLM to understand the desired format or how we approach to solve this question. And so there's a couple studies showing that giving a few examples to LLM allows it to provide better answer that aligns with our expectation. In our experimentation, we did 0, 1, and few shot, and the output looked better with just one example. So we provide one example, and here is the example of how the example reads. So we give this gene set, and we want the output to be starting with process, the name, and in parentheses is the confidence score. And we reasoning through the genes in this list, for example, the PDX1, and if there's genes that work very closely with common functions, when we group together to summarize its function, and at the end summarize overall what are the prominent biological processes involved in this gene set, and reasoning through how it's come up with the name and the confidence score. So this is the prompt that we give, and now we want to evaluate how the model performs in giving the name, giving the analysis, and giving confidence score. In order to do this, our first approach is to evaluate how much LLMs can recover known knowledge. So we here benchmark their output, their naming output, against canonical knowledge of gene set function using gene ontology. So gene ontology is a structured knowledge base for gene set with curated function annotations. So we give the LLM the same gene set and ask them to propose names and evaluate how much it aligns with the known knowledge. Here we use a panel of LLMs including GPT-4, Gemini, Mixtral, GPT 3.5, and Llama 2. And for today's challenge, you will be able to use models including Mixtral, Instruct, and Llama 2. To measure how much the LLM name recovers known knowledge, we use a metric called semantic similarity. This is a metric that evaluates the conceptual closeness between two words or phrases, not just their exact match. So what that means is that, for example, socks is closer to shoes, but it's further away from airplanes. So we use this metric to measure how close the LLM proposed name versus the human curated description in Go term. And what we found is that most of the models can return highly similar names that similar to the Go term name, whereas Llama 270B performed the worst. It has the lowest average semantic similarity. And in today's challenge, we'll be able to have a hands-on experience on how to measure the semantic similarity and evaluate the panel of LLMs yourself. So now I want to go through how we evaluate the LLM's confidence score, because it is equally important as we want to minimize the potential hallucination of LLMs. To assess the LLM's self-reflection ability, we curated a contaminated gene set by substituting half of the genes from Go gene set to other random genes, and completely randomized a gene set to generate the random gene set. Then we evaluated the distribution of confidence scores for each set. So as a reminder, we have told the model to not name a gene set if there's no coherent function for them, or if they are not confident with their names. So, and it's showing as the gray bar here in this bar chart. Apparently, GPT 3.5, Mixtral, Instruct, Llama 2 do not feel comfortable to refuse naming a gene set. In other words, they always propose something, even when there's a complete random set, which suggests that they are likely to hallucinate a name for gene set. On the other hand, GPT-4 refused to name random gene set for 90% of the time, and it gives higher confidence in a real gene set and relatively lower confidence in contaminated gene set. So we found GPT-4 are more reliable in terms of naming the gene set. So now I want to give you an example of the representative analysis from GPT-4 for this gene set I've shown in earlier slides. It has 15 protein members, and you can read the gene names here in the figure. So GPT-4 proposed the name as regulation of gene expression and cellular proliferation. The confidence it gives is medium level, and the exact score is 0.85. And what the analysis essay looked like is I've picked some text in their analysis and showing up on the slide here. It groups together some genes in the subgroup of this system and summarize what their function. For example, here those genes are involved in transcription factors, and it listed out individual functions of each gene. And here's another subgroup inside of this system. It's involved in chromatin remodeling, transcriptional regulation, and it's reasoning through individual genes again. So it can support its naming by providing those tags. And here's another example of those genes, HDAC, that are histone deacetylases, and those two proteins are also involved in modify histones. So here's so knowing that it not only provide a name for the gene set, it also provide a narrative for supporting its name. It gives more comprehensive analysis to help us researchers to better understand gene set, and for the task that we want to understand how it involved in drug response, we can add in more information in the prompt to help specify the biological context for gene set interpretation. So to sum up, I've introduced that we build an automated pipeline for gene set analysis using different LLMs, and we evaluate the ability of LLMs in recapitulating known knowledge and evaluate their self-assessment ability based on the evaluation of confidence score. And I showed the example of analysis assay, and you will be able to see them based on your experience today. And all those tasks I listed here, you will be able to have a hands-on experience with later in today's challenge. Finally, I would like to thank all those people on the slides for helping with the project and supporting the project. And please feel free to scan the QR code for the preprint of the paper, and as well as the QR code at the bottom to go to our web portal that you can try out this automated pipeline. And don't hesitate to contact me if you need you have follow-up questions for me. And I would like to thank our funding sources, especially the Bridge to AI program. Thank you.